Well, it turns out that one of the most complicated of those post-translational modifications is the attachment of sugars to those proteins. And that's the process that we call glycosylation. And by the way, you'll see that prefix glyco again and again and again throughout these lectures. That's the Greek prefix for sugar. It turns out that many of the proteins that are glycosylated are proteins attached to the membranes of cells. In fact, they reside on the surface of cells, and we call those sugar-modified proteins glycoproteins. And an example of one of those glycoproteins is shown here in cartoon form, where the protein is this blue structure that's anchored in the membrane of the cell. We also have lipids in our cell membranes that have sugars attached to them. So, for example, this cartoon illustrates what we call a glycolipid. And the sugar part of the glycoprotein or the glycolipid is what we might refer to scientifically as a glycan. Glycan is just another word for a complex sugar molecule. So because of all of these glycoproteins and glycolipids on the surface of our cells, we basically can think of our cells as having a sugar coating. And in fact, that's why on the very first slide of this lecture I had a cartoon of M&Ms. Because in many ways you can think of our cells as being like M&Ms. They're coated with sugar molecules.
Hypogammaglobulinemia is a disorder in which the body's immune system does not make antibodies, or makes a reduced amount of antibodies. Hypogammaglobulinemia has a variety of causes. The most common causes are primary immune deficiency, usually the result of a genetic mutation which prevents the immune system from making antibodies. Hypogammaglobulinemia symptoms: sinusitis, bronchitis, pneumonia, conjunctivitis, cellulitis, abdominal abscess, skin abscess, gastroenteritis.